for this work. He won the best poster at the SSU Science Symposium. So a lot of sustainability and life things are going on in the lab. And so Clayton is, in my opinion, he's a pioneer. He goes out and he has a wealth of background in building stuff. I don't know if you still have a shop for doing longboards, is that true?
Not right now, but hopefully in the near future. Yeah. Yeah.
So Clayton's able to take a project in directions that you might not guess. And he's able to take it from the beginning to the end, and we're pretty proud of the work he's done.
So I did my project on increasing the efficiency of algae carbon capture systems. And like Dr. Pulse mentioned, he was my advisor in this project. And I do want to thank John Collins, the school machinist, and Candace Gilmore, the biology technician, because they helped a lot in building the device and obtaining algae samples for this work. So I want to start out with kind of an overview of energy in the United States. So as you can see, this is a graph of electricity generation by source. And you can see that coal is by far the greatest there. It's around 39%, and then natural gas follows right after. So the vast majority of electricity in the United States right now is created by burning fossil fuels. And what that means is that every time we do that, we're releasing carbon dioxide, methane, nitrous oxide, all these greenhouse gases into the atmosphere. And you got a little bit of that from Anna's talk, but I'll mention a little bit more. And the reason that I really want to tackle electric power systems is they're the largest single source of carbon dioxide emissions in the United States. 30% of total US carbon dioxide emissions are from electricity generation. So obviously it's a lot harder to capture carbon from individual vehicles or as moving around, but with these single point sources, it's more concentrated and easier to collect. So this is a graph of carbon dioxide levels over time, and you can really see how it's following this nice cycle until around the Industrial Revolution there. We hit this extreme exponential growth. We have the highest atmospheric carbon dioxide levels in over 500,000 years. It's actually close to 650,000 years now. And these high CO2 levels cause global warming, which I think everyone knows. And also some other effects include ocean acidification. And that's really damaging to corals. It can actually dissolve their skeletons. And corals take a really long time to grow. So this is kind of a big deal for marine life too. So how do we fix this? Well, we need to capture the CO2 instead of just releasing it to the atmosphere. So there's a few different methods. These are three of the current kind of top choices for collecting this carbon dioxide. The first is amine scrubbing, and that can capture a very high amount of the carbon dioxide emissions, but it also requires a large amount of energy input. It's about 25%. So if you want to collect this 90% of the carbon dioxide, you're going to have to increase your fuel consumption by about 25% to make up for it. The other one is oxy-fuel combustion, and that means that exhaust gases are recirculated with pure oxygen, so that the end result of this generation is nearly pure CO2. You can basically compress the CO2 into canisters and then sequester it into underground caverns, which is useful in some applications. But this also requires significant energy input, between 15 and 30% depending on the system. So obviously we need a better and more efficient way to do this, which is where my research comes in. So algal systems use algae to capture this carbon dioxide. So through photosynthesis, algae take in the carbon dioxide and release oxygen, which we obviously need to breathe. You can raise them in ponds or photobioreactors, which I'll go over in a second. And this requires very small energy input. You really only need to run pumps to run air through the system. But there are a few drawbacks that I was kind of working on improving. So the two different kinds of algae carbon capture systems are open ponds, which you see on the left here, and photobioreactors on the right. And there's a few kind of advantages and disadvantages of each. Open ponds tend to have a lower startup cost. All you need is open space and algae. But you do have a lot of evaporative loss because it's an open pond in the sun all day. Photobioreactors, like you see on the right, are clear tubes where the algae grows. And this is good because you can really control all the parameters. You can control temperature, light, all those things. And it's close to the outside air, so you're not losing any water. And other types of organisms can't get into the system. So I focused on photobioreactors because you get so much more control out of it. And some of the current problems are this, you can see a biofilm. So this is the same system after 1 year. And the picture on the right, you can see that a biofilm, which is the algae sticking to the inside of the glass, has formed. And that will block the light from the algae that are still free-floating and cause them to die off. To clean this, the current method is usually to take apart the system and run sponges through and collect the algae off the sponges. And while that works, it also opens up the system to other strains of algae that can potentially eat the ones that you want to basically farm, the lipid-rich ones that are very useful. So I wanted to develop a way that these systems would be cleaned easier and not require so much maintenance. So this is my design. There's a CAD drawing on the left, and then the actual finished product is on the right, without any algae in it yet over there. And so basically, there is a power system, which you see on the top here, that runs off an Arduino microcontroller, and 2 small PV panels, solar panels, and 4 AA batteries. And that turns a gear motor, which is on the bottom there, that turns a scrubber internally, so that the system is self-cleaned every 24 hours and the algae is scraped off the walls and sinks to the bottom to be collected. The system also has sensors for light and oxygen levels in the system so that it can change the rate of airflow through the system depending on light levels or oxygen levels. So this is after 20 days. This is the second test. The first test I used a different strain of algae, which is actually the background picture here, a type called spirulina. And I switched over because it's not as rich in lipids, and it tends to be more free-floating. So for my experiment, this new kind of algae made more sense. And so I switched to a kind called nostoc, which is that kind of lime green color on the right. And it worked really well. It grew really quickly. This is after 20 days. You can barely see through the tubes anymore. And there were a few really important differences between the 2 tubes. So on the left, I have my control system, which doesn't have a scrubber and isn't monitoring oxygen or light levels. So it's just running air all the time without any changes. The one on the right has all the smart stuff added to it. So the self-cleaning bioreactor has kept the edges very clear of algae, as expected, so it's good the system's working. And one thing that I didn't realize it would do so much is create a much more free-floating culture. So all of the algae that are scraped off because of the bubbles continue to circulate, and I think that's made it a lot more efficient. And the non-cleaning bioreactor quickly developed a biofilm, and you'll see that in a second. So there's the non-cleaning reactor on the left. I cleaned it out, and then this was about 10 days later. So it grows really quickly, this thick film, and then the inside water doesn't have any free-floating algae. On the right, you can see my cleaning system running, and it'll cycle through, and you can see all the algae. So I turned it off for a few days to get this video. I let the algae grow, and then ran the system and took a video to see it scrubbing off the algae. You can see the algae gets collected on that scrubber, and then if the video is longer, when it passes by the bubbles, it gets cleaned off and the algae floats around. So to test this, whether it was really more efficient or not, I did a photosynthesis test, which means I monitored oxygen levels in each tube over time. So I fed each tube an exhaust gas from my car, actually, because that was the easiest source I had available. And so I fed them this exhaust gas for 5 minutes until all the air in the system had replaced with diesel exhaust. And then I allowed it to recirculate that air in the system for the next 10 minutes and monitor the oxygen level over time to see how quickly the algae are eating the carbon dioxide and emitting oxygen. So on the top is the control system, the one that doesn't have the scrubber, and you can see that the oxygen level decreasing down to that point where I disconnected the exhaust gas and allowed it to recirculate. And same with the bottom. And then the slopes of those recovery lines, as oxygen increases back to the original level, that shows the efficiency of each tube, how quickly photosynthesis is occurring. So it's pretty clear that the self-cleaning reactor had a much quicker recovery time, but the graph was also less uniform. I got really nice data out of the control tube, and it was a little bit bumpier in this one, even after averaging a few trials. So I'm still looking into what that could have been. So the self-cleaning reactor produced oxygen at an average rate of 2,020 parts per million per minute, circulating that exhaust gas, while the control one only produced oxygen at a rate of 970 parts per million per minute. So obviously the self-cleaning one was much more efficient. I really want to look at why that graph was so bumpy and how I could try to even that out and get maybe slightly more precise data. And so finally, the last thing that I would really like to test is to get a carbon dioxide sensor and monitor oxygen and carbon dioxide. over time to see how the levels cross each other. The reason I didn't was carbon dioxide detectors are more expensive, and the cheaper ones are generally less accurate, so the oxygen sensor really makes sense for this. And thank you. Any questions?
So you were saying that it had a quick growth rate for the second algae monocyte that you're using. At what point does that become actually hindering the process? At what point is it growing too quickly so you're not getting enough light to maintain the growth?
I would say as soon as the biofilm that grows on the edge of that tube begins to block the majority of light, the algae kind of photosynthesis occurs in kind of a logarithmic plot. So as light increases, it grows very quickly and then it kind of plateaus. So as you go further down that, the algae is going to grow less quickly. So as soon as it's blocking enough light that it makes a difference on that graph, that's when you really see a difference.
But like, do you have like some sort of time frame? I mean, like the kind of the lifetime of the tubes that you'll be using. How often will they need to be replaced?
If you have the self-cleaning system, it should last a lot longer. The other one shut down after, so the example that I had in there that I showed in the beginning, that one after 6 months, they started getting algae to die off just because of the loss of light. Yeah.
So I think an interesting metric is about the amount of return you get on kind of investment.
Yes.
So you're adding a controller, a microcontroller, you're adding hardware that could fail. But I did see a twofold, I don't know whether I can take that away, but a twofold increase,
Yeah.
at least in that. Have you done some more thinking about efficiency?
I've thought about whether this is really worth it financially. And I think that if these results, and I'm still tentative on them because these are only one style of test I'd like to do with carbon dioxide and a few other parameters. But if these results are indicative of how well this is working, I think this is definitely worth it from a cost standpoint. Because the single motor could basically run a belt system that would clean all the tubes, and you really only need one to be monitoring light and oxygen levels. So overall, as you scale this up, the control system doesn't need to be scaled up as much as well. Yeah.
Have you looked into making the biofuels yourself, or is that something that's like?
I wanted to. I didn't have really time because I ran into a few problems. I had a leak in my system and had to restart the whole thing, and it takes a while for it to grow. In graduate school, I'm going to a school that has a good biofuel research center, so I'm hoping to do some more research on that.
Yeah. And I also had a comment about your lawnmower company. You
Yeah.
should totally combine that with this and make like biofuel mopeds. Halfway there.
I like
Actually,
it.
3/4 of the way there. So yeah, it'll be awesome. I'll be calling you these people.
Thank you.